I'm going to talk to you a little bit about single domain antibodies, like Dave will mention. This picture is it took me a while to figure out what it was actually, but it's somebody taking blood from sharks. It was used in a review for in Science magazine, and because sharks are one of only two species that have these single domains. So I'm going to talk to you about that mainly, but what I thought I'd do is maybe give a little bit just about my background and how I ended up here, how I ended up working on shark antibodies, and maybe some of the lessons that we've learned by setting up a company as well. So I did my PhD in Dundee. You may know from the accent that I'm not from here. And I was working on a bug called Schistosoma mancinea. It's a parasite of tropical regions. This is an electron micrograph that we took back in the day, way back in the day. And it's a worm that lives inside. It lives inside you. The male is the dory one there, and the female is the long slender one, and they live together, and they produce eggs for something like 15 or 20 years. The eggs pass out, and then that infects a snail, and then the snail then releases these parasites that burrow through your skin, and that creates a life cycle. And that's okay, except that some of the eggs then go away somewhere, and they end up in the liver, they end up in the lungs, they end up calcifying, and that's how you can cause problems with this. But it's not a terrible killer. But I had great fun doing that, and I did a lot of other stuff in Dundee, but I also had the chance to go to a place called Woods Hole, and I don't know if anybody know where that is, but it's 2 hours down south of Boston. There was a biology parasitism course, 3 months, was fantastic. And you might recognize this guy here. This is Robin Anders. He was teaching at the course. I was a student, and that's where I said, look, this guy's science is fantastic. I'd like to come and work for you. So I got a welcome fellowship, came over to Weehai from 87 to 1990, and did a lot of great work in malaria. But one of the highlights was, of course, that Margaret Thatcher, the Prime Minister of Britain, got a chance to meet me at Weehai. And it was great. But if you look at the head of the department, Graham Mitchell's face, he's clearly terrified that I'm about to say something inappropriate. I still kept working. I still kept working there, so it was probably okay. Went back to Edinburgh, where I carried on the malaria research in David Wallaker's lab, and then came back to work in Leanne's lab, actually, as a postdoc in 1992. And that's where I've been ever since until, well, actually officially retired in December, but I've still got my lab and still going in. Mind you, Robin Anders retired something like 25 years ago, and he came in today. So he's recovered from his episode, and he was in today talking about science. But what I want to talk to you about is in the time we were there, we were getting, as we all did, NH&MRC grants and ARC grants, and the traditional way that scientists get money to study. But we had a really wonderful opportunity. Nick Hugenrad knows a lot of contacts, and we got into this cooperative research center. So I don't know if you know what that is, but it's basically the government gives a whole pile of money to a bunch of academics, some big places like CSIRO, and some companies. And they say, there you go, you've got 7 years, invent some stuff, try and sell it, and try and be self-funding after 7 years. Now, that was a bit crazy, but sometimes you can be. But as Graham Mitchell would always say, the worst place in the world to stand is between an academic and a bucket of money. And so for the first couple of years of this CRC, it was really a bit of a basket case. And it was the CRC for diagnostics. So it was QUT, it was Latrobe, it was CSIRO, and there was 3 diagnostic companies. But after a few years, we started clicking, and we started doing some really good work. It was good enough that when we went for a refund, the government said, yeah, we'll give you another 30 million. And so we ended up with like 14 years of funding to do really good research. And that's probably what enabled us to sort of set up a lot of these technologies that I'll be talking about. The it came to an end, and it was actually a very successful CRC. We spun off a company called Evogenics, which got bought over by a bigger company and then bought over by an even bigger company. And then we set up two companies, SensorLogix and Adalta. I'll talk a lot about Adalta, but just before I do that, I'll talk about the CRC for biomarker translation. That was really driven by Nick Hugenrad at La Trobe University. Again, the government gave us $30 million to try and find antibody drugs. And we got some really terrific work done, some great papers. And Nick actually developed, or Nick's group, developed a really fantastic antibody against cachexia. So that was very successful. But what I want to talk to you about is shark antibodies. So we, in the latter days of the CRC, with myself together with my colleague Stuart Nuttall, who was at the CSIRO, developed the shark antibody sort of library, we call it, and I'll explain to that in a minute. Now, when you talk about sharks, this is exactly what you think of, the great whites swimming stealthily, the predator. We went down and we used the wobbegong shark, which basically looks like a bit of a carpet. It's not terribly exciting, so I use that picture, the other picture, all the time. It's much better. But you know, just to give you some perspective, everybody sitting here has got conventional antibodies going through them. If you've been infected with anything, you'll have residual antibodies, and they're large molecularly. They're large. They've got many domains, and it's the little bits at the end here that really bind to the virus or the bacteria or whatever it is that you've been infected with. Sharks have got this other class of antibody, where instead of having two things that work together and stuck together and work as a group, they actually work as a single domain. So that means that what we do is we sort of chop that off and forget about all the rest of it, and we only use this part. And that's about 1/10 the size of a conventional antibody. But as I'll show you, hopefully, it's just as effective. It's just as tight a binder, and it can do a lot of other things that conventional antibodies can't do. For example, you'll notice that this one's got a very long we call it a CDR, but that's not important. But this loop here is actually quite long and protrudes out from the antibody base. And that means it can get into things like active sites or ligand binding sites and various other sites that antibodies can't do because they're actually working as two and they don't have that capability. It's also got extreme stability. We actually had one that you could heat up to 95 degrees and cool it down and it would still bind. It means that you can, if you have to manufacture them in E. coli rather than sort of bacteria, rather than in human cells. And that cuts down the cost. The costs of that are a lot less. And we can also tailor the half-life. Sometimes it's really nice to have a small antibody. It gets in, it does something, and then it gets cleared. But other times you need a longer half-life, and we can do that as well. And I'll show you in a second. But I'll just an interesting story. In 2017, there was a whole bunch of sort of shark incidents on the beaches along Victoria. And so the Herald Sun got in touch with me and said, "Oh, you work on sharks. This would be a good time to publish an article on shark antibodies because these sharks are getting bad press, and maybe we could have a feel-good story." So anyway, thanks for the shark. Thanks for the shark attacks. I got a bit of publicity there. This is one of my favorites, Wyeth. Wyeth was a big pharmaceutical company back in the day, and they had they were working on shark antibodies. We were working on them as well, but what they did was immunize every shark. Every time they wanted an antibody, they had to go down and immunize the shark. And that's why they've put out an advert for somebody with a degree in biology, 3 to 5 years of experience in academics, and excellent shark handling and laboratory skills. So I don't know how many people applied for that job, but I can tell you it's a very unique set of skills, that one. I always wanted to go in and just say, "Look, I'm really good in the lab." So, but why even work on shark antibodies? What's the point? Well, this slide here shows you that This is top products, and the forecast was pretty much correct in 2024. And you'll find that a lot of them are actually monoclonal antibodies. A lot of them are antibodies. 20 years ago, most of the drugs were things like statins and aspirin, small molecules. But today, the vast majority of them are actually monoclonal antibodies. So you'll see, for example, anti-PD-1, that's a checkpoint inhibitor that's been in the news a lot, very useful. This one here is actually for asthma, believe it or not. So maybe the guys downstairs are talking about that at the moment. And then there's a whole bunch of other things there. There's still a few small molecules in there. And of course, because it's 24, there was still the vaccine for COVID. But I think antibodies have proven to be a really important class. And there's something like over 100 in clinical trials at the moment for other diseases. So we thought, well, okay, maybe the sharks can do something slightly different. We won't go for those same targets. We'll do something that, maybe the traditional antibodies can't do. So what we did was we went to the Melbourne Aquarium. We got some blood. We isolated the DNA for that class of small antibodies. And we then basically have that in the freezer. And that's what we used for all our discovery work. Yeah, we didn't have to go back to the shark and inject. Sometimes the antibody came out wasn't strong enough, so we'd have to do this little thing called affinity maturation and make it stronger. But we found this a much easier way, much more convenient way to do it. But the question is, could you use? And so there was something like a billion different shark antibodies in this library, and it was all in a test tube in the freezer at La Trobe. So could we actually, but the question was, how do you find of those billion shark antibodies, how are you going to find one or two that are really important? What are you going to use? And it was at that time in 1992, I went to see a talk down here in Melbourne. I don't know if anybody remembers it, Leanne, but Greg Winter gave a fantastic talk on this thing called phage display. And a couple of years later, I went to a conference and I was sitting on the bus right next to a guy called George Smith. And he was telling me all about phage display of peptides. And then, of course, it turns out both of them in 2012 got Nobel Prizes for that. Francis Arnold did a lot of work with phage display, but in a kind of evolutionary sense. But the technique was really beautiful. Looks complicated there. Basically, what you do is you've got your billion compounds here. You put them in a little dish with the protein that you're interested in, and you just say to them, bind if you can bind. And if you let them bind, then you wash away all the other ones, and then you take those that bind and you infect them into bacteria because they're bacteriophages, they're bacterial viruses, and that propagates them, and then you do the same again. Now, if biology was 100% effective, you'd only need to do it once, but because it's not, you need to do it 3 or 4 times. But really, we call it panning. Because it's the same sort of principle. There's a few nuggets of gold in there, but there's a lot of rubbish, and you have to do that a few times in order to get rid of all the rubbish and keep the gold. So that became our standard technique that we used in the lab. So then we said, okay, well, what are we going to pick? What are we going to look for a shark antibody? What target could we do? Well, we were working on malaria with Leanne and Robin, and I'll just go back there. So this is a malaria parasite, picked a cartoon of a malaria parasite invading a red blood cell. We knew at the time that there was a particular protein that was important on the parasite called AMA1. We knew from a lot of work that Robin and others did that if you made rabbit antibodies to that and put them in to parasite cultures, it would kill them. So we thought, okay, let's see if we can get a shark antibody. We didn't know what it bound to. We didn't know what the AMA1 on the parasite bound to on the red cell in order to unlock the key, to unlock and get in. But we picked the AMA1, which is on the surface of the parasite. We panned the library, and we ended up finding a few antibodies. So, for example, here's just, I try not to show too much data, but this is an antibody that we pulled out that binds to AMA1, but doesn't bind to a whole bunch of other things. So by using this ELISA technique, you can determine, so the higher that signal, the better that binds. And that's the actual antibody that we got the crystal structure for. And you can see how small it is. It's only 118 amino acids. A normal antibody would be seven, 700, 800, even more. And when we got the co-crystal structure, we found that that big long loop actually penetrates into the sticky, the hydrophobic pocket, which the AMA1 uses to grab a hold of its receptor on the red cell. So it's like sticking chewing gum into a lock, and you can't get the key in, so the parasite can't get into the red blood cell. So that was great. Okay, it binds, but does it stop the parasite from getting in? Does it really do that? So again, this is a bit of a graph. So you add more and more of your antibody, you look to see if you can get inhibition, and you can see that one of them that we picked gives you 100% inhibition at really quite low levels of antibody. So that's fantastic. It blocks malaria parasites from invading red cells. However, when we go to a different strain of malaria, and we put the antibody in, we say, okay, what does that do? You can see that it really struggles to block invasion. So what that means is it binds to both of those, but it's really not very good at inhibiting one of them. Now that's really going to limit its use in the field. You don't want to take an antibody therapy if it's only going to kill some of the parasites and not others. It means that you're going to get malaria at some point. And what I want to do is to hold that thought in your mind. The problem with AMA1 is to try and get something that sees all of the different strains of malaria parasites. I'm just going to switch tactics now, have a drink, and talk about the company that we set up. And early on, we knew that shark antibodies are pretty cool, they're good fun, but if you inject somebody with a shark antibody, they're going to see that as foreign, and they're going to start to make antibodies to it. Now, that's probably not a big deal, except that those antibodies are going to take that antibody out of circulation. So it's going to affect the efficacy, the ability of that antibody to do its job. So what we did was we took the shark antibody, we screened the structure of that antibody, and we said, what's the closest human protein to that shark antibody? So you would think it would be one of our antibodies, but it's not. It's actually an immunoglobulin domain from our cell adhesion molecules in cells that tell cells to stick together. And just to show you, this is 3 molecules overlaid, and you can see how almost perfectly that they overlay, at least in the scaffold. The top bit is all over the place because that's the bit that binds, and that's the bit that's going to be different between antibodies. So we made a library of this time that had something like 20 billion different molecules. So essentially they're human-like shark antibodies. It means that we could actually put them into humans without having to worry too much about an immune response. So I'll quickly tell you a couple of things that Adalta's have done in the past. We collaborated with a group from WA, and we pulled out an antibody that binds to a molecule called rank ligand. That's involved in osteoporosis. And certainly in vitro, we've shown that it's able to suppress bone resorption in vitro. So again, the group in WA are looking at that, but they're academic group and they haven't really got lots of funding. Another company came to us and said, oh, do you think you could find a shark antibody or an antibody to A beta? A beta is the molecule in Alzheimer's that forms plaques. And they thought that it's not the plaques that are the problem, it's the oligomers before they become polymers and plaques. So yeah, we did it. We found an antibody that basically sees the oligomer orders of magnitude better than it does the monomer or the polymer. And there was nothing like that in the field at that point. And so we licensed that to the company. It's called Degenerex, and they're taking that forward as a potential therapy for Alzheimer's. And then Adalta at the moment is pretty focused on CAR-Ts. So these are the new sexy thing. Well, they're probably over the hill by now. But where you take T cells out of people, you put something in to target it to tumors, and then you put them back in, and therefore you can get your cells to actually target tumors. But what I want to talk to you is about the one antibody that we pulled out and we took right through into human clinical trials, and that was for pulmonary fibrosis. So fibrosis occurs, in fact, most people die eventually from some sort of a fibrosis of an organ. But really what happens is that you get an injury or an insult to an organ, you get inflammation, you get these cells that come along and try to repair it, but the repair, if the injury's continuous, the repair then overshoots. So what you end up, instead of a nice, soft, pliant tissue, you end up with calcification, you end up with scarring. If it's the lungs, it means that you can't breathe. If it's the liver, you can't, purify. If it's kidney, it's the same. And so all of these are subject to fibrosis. But there's a common molecule in fibrosis we realized at the time, and that's again called CXCR4. So again, the name's not particularly important. What's important is that if you look at normal human, is that human? Yeah, normal human lungs, you can see that there's very little staining. They're nice, they've got nice airways, nice blood vessels. But when you look at and that's called IPF, it's interstitial pulmonary fibrosis, you can see that there's massive staining of CXCR4. So your lungs are normal, they have none of this protein or very little. But when you get fibrosis in the lung, massive upregulation of this. So it suggests that that protein is important in development of fibrosis. This is also the case in the kidney. This is a human kidney, normal kidney, and look the difference between a diabetic kidney. Lots of staining of this CXCR4. And this is a mouse model that our collaborator Carol Pollock did in Sydney University. So we identified an antibody against CXCR4. And, oh yeah, hang on. And so again, I'm only going to go through a couple of these just to show you what's involved. But because it's fibrosis, you really want it to last quite a long time in the blood. So the smallness of that antibody then becomes a problem. It's going to get cleared very, very quickly by the kidneys. So we actually put a thing called an FC fragment, part of something that all our antibodies have. And that's what's able to keep this thing circulating in the blood for a long time. And we did a mouse model of fibrosis and showed that when we inject the mice, we're able to reduce fibrosis even lower than the day 8 where the mice should be when we started giving them the drug. And so that is, that we just published that recently. And this is some cool work that Louise did in her lab where you look at precision cut lung slices from humans. You can give them a cocktail, a fibrotic cocktail, and you start to get the deposition of collagen, as you can see by the green. And when you treat them with the antibody, that really sort of is significantly reduced. So we've got a whole bunch, and this is just some kidney data. Again, I won't go into this in a lot of detail, but when you've got a nice mouse kidney, when you give it a ureteral obstruction, you induce a fibrosis. You can see here the formation of vacuoles in the kidney, the increase in the staining, telling us that CXCR4 is starting to get really highly upregulated in these. When you add an antibody that doesn't bind to CXCR4, doesn't bind to anything actually in the body, you still get the same effect. But when you add our AD214, you get again a dramatic reduction of that. We never get it to zero, but it does go down. So we've published a couple of papers with Carol on this. So finally, in this part, we took AD214 and we had to make it at very large amounts, at very great expense, in order to do several monkey trials, because the FDA said, because it's a brand new therapy, you have to look in monkeys to make sure it's safe before we let you put it into people. But then we did put that into healthy volunteers. And actually we did this during COVID and we were very lucky. We picked a site in Sydney and in Adelaide, because if we'd have picked Melbourne, we wouldn't have got this study done. Now, the other thing about science is every day you go into and you do science, you're looking for some really cool, exciting results. Yeah, it's the opposite when you do a clinical trial. The last thing you want clinical trial is exciting results. Yeah, you want them to be as boring as possible. And so So that's why I've got one slide on this. It took a lot of effort from a lot of people, but really there was no dose limiting toxicities. We increased this at something like 100 milligrams per kilogram, which is a huge amount in monkeys. And we did 20 milligrams per kilogram, and we could see no effects. We looked at bloods, we looked at liver enzymes, we looked at all sorts of things. We didn't even see, we saw a little bit of anti-drug antibodies. So the body was starting to make antibodies to this, but they were extremely low titers and all to the little loops, the foreign parts of the antibody. And I think the nice thing was that because these are healthy volunteers, they don't have fibrosis. So we couldn't look to see if it affected fibrosis. But what we could look to see was that CXCR4 is on your red blood cells. And what we did was we looked to see if our antibody was binding to red blood cells. And so this is after the end of infusion. If you just look at this long thing here, this is the 20 milligrams per kilogram. So we stopped infusion here, and then we every so often we kept taking the blood cells from those people, and we looked to see if our antibody was still there. And up to about 3.5 weeks, we could still detect the antibody on the red blood cells. Now, you might think, oh, that's a bit dangerous, isn't it? Binding to your sorry, not red blood cells, your white blood cells. You might think that's a bit dangerous, but this antibody doesn't kill the cells. It just stops CXCR4, the molecule, from working. So it doesn't affect the safety of those T cells. It just stops the CXCR4 from working. But what we interpret that is to say that if we can inject 20 milligrams per kilogram or even less into people, we should be able to get it to those cells that have got CXCR4 in the fibrotic tissues, and hopefully will then prevent that. And that's where we are at the moment, trying to take that through into phase 2. We've got all the data we need for safety. We now just need to raise the money to do a phase 2, which is you get patients and you see if it actually works. The problem we haven't done that yet is because that's extremely expensive. So it probably cost us, I don't know, maybe in the region of $10 million to do that, to get everything through and then do the safety trials and the non-human primate trial. You're going to need much more than that to do phase 2. So finally, before I just go on and finish my talk back on malaria, I just want to show you, we set up the company in 2007. We finished our clinical trial in 2024. So that's a long way. In Australia, you don't have huge amounts of funding like you do in the US, for example. And so we had to reinvent ourselves a few times and pivot a few times. But what I wanted to do is just mention 2 things before I go. Oh, sorry. Oh, hang on. What I wanted to mention was this. You'll see this thing here, finalize a term sheet with a major pharmaceutical company. And that was worth something like $100 million. Now, there's this thing in biotech called biobucks. They're not real. It's like monopoly money. And what it meant was that if everything worked out perfectly, we stood to make $100 million because it was a 10-target deal and it was 10 million per target, whatever it was. That was with Bayer Pharmaceuticals. We signed the deal. Their lawyer signed the deal on Friday and texted us, emailed us, and said, you can quietly open the champagne. I have just signed it. So we were ecstatic that whole weekend until Monday when we got another email saying there's a new sheriff in town. So they got a new head of biology and he was putting a stop to any deal that he hadn't done. And so we couldn't do anything. We were a small Australian biotech company. They were a large German company with tons of money. And so we just had to suck it up. The other one is I guess it was nice that it happened when we were young because we were not expecting too much. But this one here was a few years later. And this was Novozymes, a Danish company who had a subsidiary in Adelaide. They came over and talked to us for maybe 6 months. And then eventually they sent lawyers and various people over. It must have cost them at least $5 million just to do the due diligence on all of that. And they said, okay, let's do it. They signed, we had the deal all ready to go. The guys from Adelaide went over to Denmark, were sitting there waiting, and the guys came out and said, sorry, global financial crisis means that we're not going ahead with any deals. And not only that, the Novozymes facility in Adelaide is closing down as well. So when I say highs and lows, that's exactly what I mean. And we've had, there's people in the audience here that work for Adalta, and we've had to let them go because we had no money at some point, and then suddenly we get money and we employ other people. So it's not easy being green, but it's the highs are really good, but the lows kind of suck. Let's just jump back to malaria. I've only got another 10 minutes. Malaria and influenza. So we've been working on those two for a while. They're not really the same, except they're both pathogens that invade your cells. You know all about malaria because I've explained that, but essentially influenza does the same. And parasite uses AMA1, among other proteins, of course, but one of the proteins that uses AMA1 that I showed you, that we got a shark antibody to, but it didn't see all AMA1s from all parasites. Of course, the influenza virus uses hemagglutinin, but it's got the same problem. They're all basically, extremely polymorphic. This is AMA1 showing you all the different amino acids in one part of the AMA1 protein. This is the hemagglutinin showing you all the regions here that can mutate. That's why we have to get a new vaccine every year for influenza, because next year there'll be a different strain. Your antibodies won't see it because the parasite will have mutated. So it's a little bit like, they're all, it's all malaria, it's all influenza. These are all finches, but they're all different colors and different. So what we have to do is try and get something that sees all of them. And so that's what we did. We took AMA1. So we took AMA1 from one strain of parasite and another strain of parasite. So this time what Dimitu did in my lab, he was a PhD student and he wanted to work on malaria. I said, okay, sort this out, solve this problem once and for all. So he took AMA1 from 3D7 strain. Again, the strain names don't matter. But what's important about these two strains is that they're quite far apart in evolution. Okay? So the thinking here is, if we take 3D7 and we pan on that just 3 times, so we do 3 rounds of panning, so what comes out must bind to 3D7, and then we put those antibodies on W2-MEF and do 3 rounds of panning on that, then anything that comes out the bottom has to see both of those, otherwise we would never find it. And hopefully if we choose them from very far apart in evolution, it might see all the others as well. And I'll go through that. And that's what we did. This is just a summary of a lot of the data that he did. And the really dark red spots are strong binding to AMA1. So you can see that 2 of them, WD33 and WD34, WD40 was taken. That you can see that they're binding to not just 3D7 and W2-MEF, which we used, but to 7G8, FVO, and HB3. But the question is, okay, so of those 2 antibodies, do they block the binding of AMA1 to the receptor? Because now you know what the receptor is. It's a molecule called RON2. And you can do that in the laboratory, and you can see that WD33 has no effect whatsoever on the binding of AMA1 to its receptor, but WD34 inhibits it beautifully, a nice dose-dependent inhibition of AMA1 binding to its receptor RON2. This is showing you that, just another way of showing how the antibodies bind. So if you get a nice band here, AMA1's got 2 bands, you see that it binds to all of the strains of Plasmodium falciparum. But what we found was that it binds to Knowlesii and Berghii, as well as 2 other strains. I don't know, anybody heard of Toxoplasma? So toxoplasma is a big problem for immune-compromised individuals or pregnant women. And Babesia is a big problem in cattle, but it's becoming a massive problem in humans, actually, particularly in the US. In fact, some of the drugs that are on sale for malaria, they're getting more sales against Babesia in the US than for malaria. And so what it meant was this not only saw the different strains of Plasmodium falciparum malaria, but all sorts of things, because they're all called apicomplexes. They all use AMA1. And so we thought, oh, this is pretty good. So let's find out where it's binding on AMA1. And again, DIMA2, in collaboration with Mark Vanschool and a group at La Trobe, found out that the antibody binds to this region here that I've coloured in. And what's interesting is that that green bit there is the hydrophobic region of AMA1 that binds to the receptor. Okay, so you can see again, this antibody will block that completely by lying across it. But importantly, this is the main region of AMA1 that's very polymorphic. It's got lots of mutations in that region. This antibody binds way, way, way away from that. So that explains probably why it's able to bind to all the different AMA1 strains. And we've never seen a molecule like this, and I've got shark antibodies and peptides and rabbit antibodies and mouse monoclonals against AMA1, and we've never seen a reagent quite like this. We showed that, again, I won't get into the nitty gritty, but this is actually blocking parasite growth. So that you can see that the more antibody you add, the better it inhibits the growth of parasites. It's able to block all of the parasites, including P. vivax and P. nosei. And interestingly, it's also able to block the liver stages as well. We give this to Justin Bodie at WeHi. He's got the mosquitoes and everything running so that he can, and the liver stage is where the mosquito bites you and the sporozoite gets in and goes to the liver. And what's incredible about this is that AMA1 is involved in getting into the red blood cell, and it's involved in getting into the liver stages. So if we use this as an immunotherapy, we've got a double whammy. If we can stop it getting into the liver, and if it does get into the liver and bypasses it, we should be able to inhibit it also at the red blood cell stage. We gave some of this antibody to Chris Tonkin at WeHi, who's got toxoplasma growing in the laboratory. He showed that it was able to block toxoplasma in vitro. And we gave also some up to Daniel Stanisic and Michael Good's lab in Griffith University on the Gold Coast, and showed that it could at least reduce the parasites in another strain of Plasmodium in mice. So this is an actual mouse model of Plasmodium infection. And you can see that if you don't give them anything, they get massive parasitemias, lots of parasites in the blood. And if you add an antibody that doesn't have any effect, you still get very high parasites. But it's dramatically reduced. We're still doing a lot of work to get the dosing right to try and see if we can knock this down to zero. But what's interesting is that they did a babesia. They've got a mouse babesia model. And again, we get inhibition, dramatic suppression of parasites in the blood in these mice. So I said, okay, that's fantastic, Dimitri, well done. We published that. I said, what about doing it with something else? So he did the same thing in malaria. He took H1 hemagglutinin, which is from group 1, and H3 hemagglutinin, which is from group 2. And he did the same sort of panel 3 times on H1, 3 times on H3, and asked the question, could it bind to all of them? And, just to show you that I'm giving you some real results here. This is what we're looking for. Beautiful. This antibody binds to both of them. The red and the blue overlap. So does this one. But this person here did not. It bound to one of them, but not the other. Actually, it's not true. It just binds really low to the other one. So they still come through, even though your panning is supposed to be work. But what we showed was that, for example, this class of antibody is able to bind to all the H1 strains that we have. And this was done with collaboration with the guys at the Peter Doherty, Patrick Reading's group in the Peter Doherty. And it was able to bind to H3, H5, and even influenza B. So our idea here was that this is a pandemic preparedness, that if we have an influenza pandemic, we would have an immunotherapy that you could inject in immediately, because you don't need to know what strain the influenza is, and it would be there until you could get a vaccine. So, for example, particularly for frontline workers, for example, that have to work away in hospitals, nurses, first responders, that have to work in a pandemic setting before there's a vaccine. And I think this is my last slide, or it's certainly close to it. So what we've done is developed this antibody that's able to see all of the different strains of Plasmodium. We've got the patent for that, so we're trying to find funding now to get it to the stage where it's ready to go into the clinic. And it's really fantastic here. There's a guy called James McCarthy who's got a human challenge model of malaria, which means that you can actually do a phase 1 and a phase 2 together in the same trial here in Melbourne. And you can do that for really not a lot of money. One of my colleagues did a dengue trial for less than $5 million. So I explained to you before about how much you need to do these vast clinical trials. That this human challenge model in infectious diseases is really a game changer, I think. But there's 2 papers out showing you that these are monoclonal antibodies, traditional monoclonal antibodies that are being taken through the clinic. But again, I'd highlight that those are only to the sporozoite stage of the vaccine, and they don't see Plasmodium vivax, for example. So we think we've got a slight edge on some of these that are coming through. And this is just, I tried to mention everybody as I've gone through, or a lot of people. There's a lot of people, for example, in the audience that have done work that I haven't. But these are the guys that have contributed to the work recently. And it's really been driven by Dimitu with a lot of effort from Rob and others. So thanks very much for listening.
So let's have some questions, and I'll take Chairman's privilege and ask you the first one. There's clearly a lot of advantages of having a small molecule versus a big antibody. But the problem with antibodies is you've got to inject them. So is there some way that you can deliver these without needles?
Not really. No, there isn't. I'd love to say there was. There isn't. I mean, the I-bodies themselves and the shark antibodies, we did a lot of work early in the day. I mentioned that they're very stable. So you can, some of them you can heat up, even to 90 degrees, cool them down and they still work. And we also put them into simulated gastric fluid and they were fine. They didn't get chopped up. And we did put them into scrapings from rat stomachs and intestines. And again, they were pretty stable. But they're not going to get through. They're not going to get through the gut, and they're not going to get into the blood. That's the problem. So perhaps if it was for Helicobacter or something, that might work. Although we'd have to bind in that pH as well. So the answer is no. But what we're doing at the moment for the fibrosis one is that it is an infusion, and that's just the easiest way, the most accepted way. But we can reformulate that to get a subcutaneous. So then it becomes like an EpiPen. And then I think compliance then becomes a lot less. And so I think that's the benefit is that you have an EpiPen with something with a reasonably long half-life